Happy Thanksgiving week. Happy the exam is over week. I can see where things rank in your priorities. How was the exam? You liked the exam? No? Okay. All right. They're never going to have 100%. So, thoughts about the exam? Hard? Okay, the question, when you get it back. I'm hopeful, fingers crossed, we'll have it available sometime on Wednesday. That's our hope. I think it's possible, but it also depends on their TA's schedules as well. They have things they have to do, but my hope is Wednesday. No comments? You liked the extra credit question, okay? That was a sneaky thing, wasn't it? Yes? Yes? Yeah. So you're thinking I should make it available Wednesday and then cut off the all comments by, what, Saturday? You think that's a good idea? I'm a reasonable person, okay? So maybe we'll count day one as Monday. How about that? Okay. And please don't come bug him in the office, but it probably will be Wednesday, and you will get an email from me when it's available. So, if not, announced in class. Speaking of class, as you probably know, I've announced earlier, but I'll repeat myself. No recitations this week. TAs are available if you want to go ask them questions and so forth, but no recitations this week. And if any of you are around on Thursday and you don't have plans, come over to my house. Serious, my wife and I put together a big turkey dinner every year, so if you're interested in that, let me know. Get overwhelmed with 400 people in my house. Kind of cool. But seriously, do let me know. Okay. We are plotting our way through sugar metabolism, and we're spending a fair amount of time on sugar metabolism because sugar metabolism is pretty critical for our bodies. We are pretty much done with glycolysis. Today we're going to talk about regulation of glycolysis. We're going to talk about the synthesis of glucose, which is gluconeogenesis. And we're going to talk about how these impact things like cancer and weight gain. Okay, some very serious topics. So without much further ado, I'm going to dive into those. So let me first talk about regulation a little bit. Okay, and then I'm going to come back and talk about other sugars. So I briefly, and by the way, I'm going to talk about regulation in two senses. First, I'm going to talk about regulation with respect to glycolysis. And after I've covered gluconeogenesis, then I'll come back and I'll talk about regulation again with a view of glycolysis and gluconeogenesis. And the reason for doing that is to make you aware of the way that these two pathways are interregulated. Okay? So the first things I'll talk about regulation are pretty straightforward. Okay? One relates to this enzyme that I said was the most important enzyme for controlling the glycolysis pathway, and it's the Phosphofructokinase enzyme, or as we will call it, PFK1. Okay? PFK1 is an enzyme that catalyzes the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, and this reaction shows, this graph shows some interesting things. Okay, we can see the reaction velocity as a function of the enzyme being in either high ATP or low ATP concentrations, and this, at first glance, goes, oh, well, it makes sense. At high ATP, the enzyme is inhibited, and it's probably in the T state, and that makes good sense. But then, if you think about it, you scratch your head and you say, but isn't ATP one of the substrates of this enzyme? For which I would give you a gold star and say, yes, ATP is, in fact, one of the substrates of this enzyme. And then we have to say, well, how is it that high levels of a substrate is actually inhibiting this enzyme? That doesn't make any sense. So that's my question to you first. How can the high levels of a substrate inhibit an enzyme? That's not something we've seen before. Yes? Does too much ATP prevent fructose 6-phosphate from binding? Indirectly, yes, but that's not the answer to the question. Yes? Does that have to do with having too much energy? No. Although that is a fact, in fact, that if you have too much energy, you don't want glycolysis going, but it doesn't explain how the enzyme works. You actually could figure this one out. Maybe I should let you figure it out. What do you think?
Allosteric binding.
Allosteric binding, okay, but how does that affect this? Your answer is right, but I'm trying to extract more information out of you.
By binding to the allosteric site, it prevents the enzyme from grabbing more fructose 6-phosphate and catalyzing it.
But ATP is a substrate. Back here. It is allosterically regulated by ATP, but ATP is a substrate. What's that? By what? ADP. It actually is regulated by ADP, but that's not the cause of this phenomenon. Yes? Too many people in the room just get in each other's way. That happens a lot too. The answer is no. Okay? This is a puzzling enzyme. I'm going to give you the answer. The answer is that there is, and this is what I'm trying to extract out of him over here, there's a separate allosteric site for binding ATP from the active site which binds ATP. Okay? So you have two places on the enzyme that can bind ATP. You have an allosteric site and you have an active site. You'll actually see this next term when you talk about ribonucleotide reductase. Okay? Now, if I were to ask you on an exam, which site has the greater affinity for ATP, which one would you say? The active site or the allosteric site? Allosteric. Somebody bold jumped forward and said allosteric. The answer is wrong, unfortunately. Okay? It's the active site because you want this enzyme to be able to work in low levels of ATP. Because remember, in low levels of energy, you want this reaction to go. So you need to have the active site have a high binding affinity for ATP, and you want the allosteric site to have a low binding affinity for ATP, such that it only kicks in when the ATP levels of the enzyme, of the reaction, are high. Okay? So very, very interesting enzyme. We'll see this enzyme is interesting in other ways as well. Okay? Probably the most important regulator of the enzyme is not ATP, it's not ADP, and it's not other things that I'll show you later, but in fact, it's a molecule called fructose 2,6-bisphosphate. That sounds like an intermediate of glycolysis, and it's not. Okay? It's structurally similar, but it's not an intermediate of glycolysis. It's made from fructose 6-phosphate by a different enzyme. Now, for reasons that are never very clear to me, students always get confused about this, and they get worried about this intermediate. Where does it come from? Nobody says, where does ATP come from? Where does ADP come from? Where does pyruvate come from? But this one, they'll say, where does it come from? It doesn't matter. It really doesn't matter. It's made by an enzyme. The cell makes this molecule, and we'll see when we talk about the regulation of gluconeogenesis how, when, and why it makes it. All right? But the reason it's making this molecule is to help control glycolysis and gluconeogenesis. Okay? This molecule activates PFK, and it activates it very strongly, as you can see. Very tiny amounts of this molecule have a dramatic effect on the enzyme's velocity. Okay? Very dramatic effect on the enzyme's velocity. Okay? We will see later that this same molecule inhibits the equivalent enzyme in gluconeogenesis. It's a phenomenon we refer to as reciprocal regulation, and I'll define that term for you later. Okay. This is what it looks like, and you can probably figure that out yourselves, given that you already know the structure of fructose. You put phosphates on carbons 2, and 6, and you have it. Okay. Let's not talk about that. And let's not talk about that. But let's think, well, actually, I want to come back, and that figure I find confuses people a little bit. So the most important enzyme for regulation is phosphofructokinase. There are 2 other enzymes that are regulated in the pathway. The first is hexokinase, and I should mention that one. It's not shown in my outline. Hexokinase is regulated. Its regulation is at the level of what we call substrate-level regulation. Okay? It's not an allosteric regulatory mechanism. And it's an odd mechanism in that the concentration of the substrate actually does affect the enzyme, such that the product, as it builds up, inhibits the enzyme. Okay? We won't really say much about the regulation of hexokinase other than to note that it is a regulated enzyme for the pathway. The third enzyme of the pathway is the one I mentioned earlier that's odd. It's the last enzyme of the reaction. Okay? It's the last enzyme of the reaction. It's pyruvate kinase. I still have phosphofructokinase in my head. It's pyruvate kinase. And pyruvate kinase turns out to be regulated by 2 different mechanisms. One mechanism is allosteric, as you've seen before. In allosteric regulation, ATP turns it off, and this makes perfect sense. If we have a lot of ATP, we don't want to be making pyruvate. Alanine also inhibits it, and that might seem a little odd. Why does an amino acid affect that? Well, it turns out that pyruvate is very easily converted into alanine. They're very easily interconverted. That means that there's a lot of alanine, there's probably a lot of pyruvate sitting around. Again, both of these now are telling the cell, hey, we've got plenty of energy. We've got plenty of intermediates. We don't need to be running glycolysis. We don't need to be lighting our furnace in the summertime. Earlier, I talked about how fructose 1,6-bisphosphate activated this enzyme by this mechanism I called feed-forward activation. I won't go through that again, but check back on the earlier notes for that. Fructose 1,6-bisphosphate activated this enzyme. The second level of regulation of this enzyme is covalent modification. And the covalent modification, you can see, is regulated by phosphorylation, dephosphorylation. Okay? One of the enzymes that can put a phosphate onto this enzyme is our friend protein kinase A. Enzyme that takes the phosphate off is known as phosphoprotein phosphatase. So the most active enzyme will be dephosphorylated with fructose 1,6-bisphosphate there. We'll have intermediate ranges of activity in other states. Okay? Why is this enzyme regulated? Well, as we will see when we talk about gluconeogenesis, gluconeogenesis has to sidestep this enzyme because remember this enzyme catalyzed the Big Bang, right? The Big Bang meaning it was extremely energetically favorable in the forward direction. Okay? Extremely energetically favorable in the forward direction. What does that mean? It means that if I have PEP and I have an active enzyme, I am going to convert it to pyruvate, no ifs, ands, ors, or buts. I'm going to have virtually no PEP left. Why do I care about that? We'll see that in the pathway of gluconeogenesis that we do 2 steps, going from pyruvate to something called oxaloacetate to PEP. Imagine that this, and by the way, that takes 2 triphosphates to go, those 2 reactions, to oxaloacetate and to PEP. Imagine the cell has blown 2 triphosphates in getting back to PEP because it's trying to make glucose, and this enzyme is still active. What's going to happen? Well, this PEP is never going to make it back to glucose because it's going to get converted right back to pyruvate if it's on. So being able to regulate this enzyme is essential for the pathway of gluconeogenesis. And I'll talk about that when I talk about gluconeogenesis. So, glycolysis is odd. The last enzyme is regulated, and it's regulated because of that giant energy advantage of the Big Bang that I talked about earlier. Okay. Let's talk about some other sugars. Okay? This figure shows us something interesting, and that what it's showing us is that glycolysis is really a pathway for metabolism of a variety of sugars. Galactose can readily be converted into glucose 6-phosphate. Fructose can readily be converted into fructose 6-phosphate. And both of these, of course, can be metabolized in the pathway of glycolysis. So other sugars can enter the pathway and be metabolized. Let's take a look at how that happens. The first one I want to talk about is fructose. Fructose is really interesting in this respect. Okay? I'll tell you why in a minute. Here's fructose. There's an enzyme called fructokinase that we have in our cells that will convert fructose into fructose 1-phosphate. Okay? Fructokinase catalyzes that reaction. There's another enzyme in our cells called fructose 1-phosphate aldolase. What did aldolase do in the glycolysis pathway? It split a 6-carbon molecule into 2 3-carbon molecules. Fructose 1-phosphate aldolase splits this 6-carbon molecule into 2 3-carbon molecules. One you've seen before, dihydroxyacetone phosphate. That's an intermediate in glycolysis. And here's another molecule called glyceraldehyde. And you've heard something like glyceraldehyde before because in glycolysis we had an intermediate called glyceraldehyde 3-phosphate. Looky here. Triose kinase will put a phosphate onto glyceraldehyde, and we've got glyceraldehyde 3-phosphate. Fine and dandy. We have metabolized fructose. We have gotten it into glycolysis. Everything is fine and dandy. And I'll tell you what you see on the screen right here is one of the reasons that Americans are fat. Now, that's another question I have for you today. Why does this, what's on the screen, tell me why Americans are fat? Okay? Because we started with fructose and we end up with 2 glycolysis intermediates. Why is that any different than starting with glucose? Well, the answer to that question starts with something that we decided to do back about 30 years ago. About 30 years ago, we decided that there were too much duties on bringing sugar in the form of sucrose into this country. So instead of paying those duties on sucrose, we said, hey, we've got plenty of fructose around in the form of high fructose corn syrup. And it's sweet. Let's use high fructose corn syrup to sweeten things. And so we started doing that. The fructose consumption in this country went way up. A bunch of idiots that went to health food stores in those days saw that they could buy pure fructose. It's the natural nature's sugar, and they started buying pure fructose to sweeten their drinks because, hey, somebody at the store told them it was really good for you. I hope I'm going to convince you otherwise today. So what's wrong with it? Why is this making Americans fat? Okay? We start with fructose, we end up with the glycolysis intermediates. Why is this doing this? Okay? Any thoughts? What's that? It's more direct than breaking down glucose. What does that mean? This happens faster than breaking down glucose. No. Requires less energy. No. Okay? What's that? You thought energy? Okay? Up here. What? Because what? It skips a regulatory step. It skips the most important regulatory step, which is PFK. Now, if we skip a regulatory step, what do regulatory steps do? Regulatory steps are there to stop things from going forward. Okay? We're bypassing the regulatory step, and now we're down into the 3-carbon molecules. And when we start making 3-carbon molecules, folks, we are going to force the production of pyruvate. We are going to force the production of pyruvate. Pyruvate is an end product of glycolysis. Pyruvate gets broken down to acetyl-CoA in the mitochondrion. And if we didn't already have too much energy, it would get metabolized to carbon dioxide. But if we have too much energy because we have too much of this stuff coming through, our citric acid cycle stops, as you will see next term. When the citric acid cycle stops, acetyl-CoA concentrations start to increase. And when acetyl-CoA concentrations increase, you got to know that acetyl-CoA is a precursor of fatty acids. Bang. This is the reason Americans are getting fat. Okay? Now, people say, well, what if you have sucrose? Don't you have fructose and glucose? So you're already getting, if you're eating sucrose, why is that better than eating this, than getting this? Okay? The answer is high carbs are bad, period. Okay? High carbs are bad, period, because high carbs will do exactly what you see here, starting, okay, with the fructose 6-phosphate reaction. Okay? High carbs are going to force forwards the production of pyruvate. Okay? Now, there are fad diets, there's all kinds of fad diets, et cetera, et cetera. But as a general principle, reducing carb intake has been shown in many studies to be good for you, not harmful for you. Okay? Reducing carb intake will have an effect of reducing the synthesis of fatty acids and reducing the obesity happening as a result of that. Questions about this? Yes, Garrett.
Before we had high fructose corn syrup, what was like the normal intake of fructose?
Before the advent of high fructose corn syrup, what was the normal intake of fructose? I couldn't give you a number if that's what you're looking for. I don't.
Like, on average, like compared to now, it's greatly increased.
Okay, so there's considerably more carbs that we're taking in the form of both fructose and carbs in general. So though I'm saying that fructose is the villain, it's actually carbs, high carbs, that are the villain. Yes, Deidre. Yep. Yeah, a common question I get. Fructose is a common component of fruit juices, which is one of the reasons that the health food stores were telling everybody, eat all this fructose because it's good for you, right? It's what we have in apple juice. It's what we have in grape juice, et cetera, et cetera. What fruit juices tend to have with them are things that regulate metabolism of sugars. They actually have some inhibitory molecules in their fruit juice that affects your metabolism of sugar. Okay? Now, while I wouldn't go out and recommend drinking a gallon of apple juice a day, I will tell you that drinking fruit juices and eating fruit is better for you than drinking high fructose corn syrup that doesn't have those things. Okay? Up top, yeah.
What about like complex carbohydrates?
Yep. Yeah, what about complex carbohydrates compared to simple carbohydrates? Again, numerous studies have shown that complex carbohydrates have a very low, what we call glycemic index. The glycemic index is a measure of how rapidly the carbs make it into your bloodstream. And so complex carbohydrates have a low glycemic index. They don't make it in so fast and have much less of an impact than if you're having sugar. So,
Does that mean it's regulated, or does that mean it can't?
does that mean it's regulated? Okay, so the issue of regulation is one of being overwhelmed. So if you have something that has a high glycemic index, the likelihood you're going to overwhelm is much greater than if you have something that has a low glycemic index. Okay? Question down here.
Yeah, so when you drink fruit juice, does the concentrate they still contain those inhibitory molecules or not?
When you drink what?
Fruit juice from concentrate.
When you drink fruit juice from concentrate, does it still have those inhibitory molecules? I have no idea. Yep. Okay, so people with diabetes who are eating candy bars to up their blood glucose, are they getting fructose that's inhibitory or not? And again, I can't give medical advice, so I can't answer that question. Garrett? Why don't we have regulatory inhibitors for fructose? It wouldn't be in our interest to do it. Our bodies are here to reproduce. They're not here to live to be 80 years old. No, seriously. So we're here to get as much energy as we can, to reproduce as much as we can, and then die as quickly as we can. That's a sad story, but it's a fact of nature, folks. Yeah, one last question. Was it favorable for the cavemen to be able to process fructose? It was favorable for you today to be able to process fructose. It's just not favorable for you to live to be 80 years old. Okay? All right. Okay. Now, let's talk about galactose metabolism. There's something kind of cool there, too. Galactose we get in our diet from milk. Lactose, you remember, is what's known as milk sugar. And lactose breaks down into galactose and glucose. So there's a great source of galactose for you. Okay? What I'm telling you today has nothing to do with lactose intolerance, although the source of lactose might arise from lactose intolerance. Once you have galactose in a free form in the body, there's an enzyme called galactokinase that will put a phosphate, again, onto position 1 and make something called galactose 1-phosphate. Okay? Using energy from ATP, as you've seen before. Okay? Galactose 1-phosphate in the body is normally converted into glucose 1-phosphate. I'm going to show you how that happens, and though it seems very complicated, it's actually not. All right? Galactose 1-phosphate swaps glucose with something called UDP glucose. We talked about it before. We'll talk about it again when I talk about glycogen metabolism. But UDP glucose is a molecule we have sitting around in our cells. It's actually a precursor in the synthesis of glycogen. These enzyme names you don't need to worry about. But there's an enzyme that takes the glucose off and puts the galactose on. So we're replacing the glucose 1-phosphate on here with galactose 1-phosphate. So the product of this reaction is UDP galactose and glucose 1-phosphate. We'll see soon that glucose 1-phosphate is readily converted into glucose 6-phosphate, and then that can be burned in glycolysis. Okay? So what I've shown you is that we've got galactose 1-phosphate that is releasing a glucose which goes into glycolysis. What about the galactose? The galactose here in the UDP galactose gets simply converted into glucose in another reaction. All right? And that is our starting material right there. So what this is a cycle. Every time the cycle turns, one molecule of glucose 1-phosphate is produced. Okay? So in essence, we are converting galactose 1-phosphate into glucose 1-phosphate. And glucose 1-phosphate can be used in glycolysis. It can also be used to make glycogen. Okay? Now, that's all fine and dandy, but if we have enzymes, such as this enzyme or this enzyme, that get gummed up, that is, if they get, a person has a mutation, or if we overload this system, what will happen is we will accumulate galactose 1-phosphate. Everybody understand that? So if we have a person that has either deficient enzyme or we have people who have overloaded the system, they will accumulate galactose 1-phosphate. Accumulating galactose 1-phosphate is bad because galactose 1-phosphate is the product of this reaction. And if this guy accumulates, what's going to accumulate? Galactose. And when galactose accumulates, okay, it gets converted by another enzyme that sees galactose as a poison, because remember, it's an aldose, just like glucose is. When galactose accumulates, the cell says, that's a poison, I'm going to convert it into galactitol. And galactitol has a problem. Galactitol will crystallize in the lens of your eye and start to form a cataract. Okay? So one source, and there are many sources of getting cataracts, but one source of getting cataracts is accumulation of galactose and then the subsequent formation of galactitol. It's a reduction. You see, we're starting with an aldehyde, we're going to an alcohol. And if you want really good evidence that cells consider aldoses to be poisons, here's a really good example. The cell is saying, I don't want that thing around because it's going to be problematic. I'm going to convert it into a much less reactive molecule, which has an alcohol group instead of an aldehyde group. Okay, questions about that? Yes? Would a person who's lactose intolerant be less prone to having cataracts? I don't know the answer to that question. On the surface, it would seem like, yes, that would be the case, because you would guess that their galactose from the breakdown of lactose would be reduced. There are probably other sources of galactose in the diet besides the breakdown of lactose. So you might eat things, for example, that have free galactose in them. And I doubt that I imagine those would probably contribute as much as the lactase enzyme would. So as I said, I don't know the answer to the question, but I suspect it's not a significant difference. I'm not a nutritionist. I don't know what else has galactose in it. But again, any of the sugars you'll find in a variety of places. Yeah. So this buildup happens. There's a couple ways it can happen, as I said. One is through having a deficient enzyme, but the other is overloading the enzyme system that I described to you. So a large consumption of galactose could also have that effect. Okay. Well, we're moving through things. I want to talk about glucose transport proteins. Okay? Remember when I talked about the insulin signaling pathway? I said that the insulin signaling pathway worked because it favored the movement of proteins I called gluts up to the cell surface, the cell membrane, and these gluts allowed the input of glucose to occur. Here are some gluts. Okay? You can see that they have specific locations in the body. Glucose transporters are important. Glucose transporters become particularly important when we consider a tumor. Okay? Here's a tumor. A tumor forms, it's undifferentiated tissue. It forms at a location in the body. And tumors frequently will have what we think about as a very rapid metabolism, meaning that they turn over very rapidly. They divide more rapidly. If they're dividing more rapidly, they must therefore need more energy more rapidly than a normal cell. Okay? Well, energy is important because if we think about it, energy is needed for making nucleotides, for doing DNA replication, for making proteins, doing all these things. So tumor cells need a lot of energy. I've told you that the way that cells get the most energy is by breaking down glucose in the presence of oxygen. Cells get their maximum amount of energy by breaking down glucose in the presence of oxygen, because in the presence of oxygen, each molecule of glucose can give 38 ATPs. In the absence of glucose, each glucose gives 2 ATPs. It's in the interest of a cell to get as much oxygen as it can. And one of the things that we see of tumor cells is that they frequently stimulate the growth of blood vessels. What do blood vessels do? They bring blood, and what does blood bring? Oxygen. So if we think about ways to starve tumor cells, one of the strategies is to stop the development of new blood vessels, because if we stop the development of new blood vessels, the tumor may starve itself to death. And there are actually some chemotherapy strategies out there that people have employed right now to make that happen. Okay? Not all tumors have this phenomenon, though. There are many tumors that go on and exist, okay, even without an increased blood flow. Most tumors will be discovered are what we call hypoxic, meaning they have low levels of oxygen. Hypoxic. Cells that have low levels of oxygen occur in our body all the time. Whenever you go exercising, you're making hypoxic muscle cells. The body has to respond to that because, let's face it, hypoxia is a fact of life. Okay? So we already have built into our cells something called HIF-1. HIF-1, hypoxia induction factor, is a protein. You saw it on the previous slide. Under conditions of hypoxia, HIF-1 is activated, and HIF-1 activation stimulates several things, including blood vessel growth. Okay? What's interesting are the other things that it stimulates the production of. So let me tell you what HIF-1 is. HIF-1 is what we call a transcription factor. A transcription factor. Transcription factors are proteins that activate the transcription of other proteins. So HIF-1 is a protein that's activating the transcription of other proteins. Let's imagine that I've got this tumor here, and even though it's got blood vessel growth coming to it, it's still fairly hypoxic. It's going to have low oxygen concentration. What proteins do you suppose would be good for this tumor cell to induce the production of? I'll tell you some. Yes? Glycolysis enzymes. Excellent. I think that's worth another point on an exam. Okay? If we look at cells that have HIF-1 active, look at the proteins that get stimulated. Gluts, no surprise. The more gluts you put on the surface, the more glucose you can bring in. And if you have no oxygen, you got to bring in 19 times more oxygen. 38 divided by 2, 19 times. Okay? The more glucose you bring in, the more enzymes you've got to have to handle it, because remember, we don't want to have saturating amounts necessarily, because we're going to have enzymes, or we're not going to be able to produce as much as we want. So we make more enzymes so we can make more glycolysis. And look at the enzymes that's made. Hexokinase, phosphofructokinase, aldolase. All of these are enzymes of glycolysis. Okay? Very important consideration. So, if you can think of ways to block HIF-1 action, you might have another strategy for chemotherapy. Okay, questions about that? Okay. Well, I want to turn our attention in the last few minutes that we've got of the class to talk about glucose synthesis. Okay? Glucose synthesis in our body is absolutely critical. Just like glucose breakdown is absolutely critical, so too do we have to be able to make glucose, because if we can't do that, the body's going to be in trouble. And we'll see why that happens in just a little bit. Let's start here. Okay? First of all, I should tell you, if I haven't already told you, I think I have, but I'll tell you that glycolysis, all the reactions of glycolysis occur in the cytoplasm of the cell. All of the reactions of glycolysis occur in the cytoplasm of the cell. All of the reactions of gluconeogenesis, except two, occur in the cytoplasm of the cell. One of the things we see with gluconeogenesis is that 7 of the glycolysis enzymes are used in the pathway of gluconeogenesis. Remember, glycolysis had 10 enzymes. Gluconeogenesis uses 7 of those enzymes to catalyze reactions that are the reverse of the glycolysis reactions. Remember, enzymes catalyze reversible reactions. So, all the enzymes shown in blue here are used in both glycolysis and gluconeogenesis. Enolase, you can see all these things. So, when we are going from glycolysis direction, we're starting at the top and we're moving down. When we do the gluconeogenesis, we are starting at the bottom and moving up, reversing the pathways. Now, why don't we use all 10 in reverse? And I think you know the answer to that question. The answer to that question is that we have 3 big energy barriers we have to get over, the first of which is the Big Bang. The Big Bang, remember I said, was so favored in that forward direction of PEP going to pyruvate that we're not going to back it up. We're just not going to back up that reaction. From an energetic perspective, it's almost impossible to back up the pyruvate kinase reaction. So cells don't try to do the same enzyme in reverse. Instead, the cell does what we call a 2-step. By the way, I should also point out, gluconeogenesis is not present. It does not occur in all the cells of our body. Gluconeogenesis occurs primarily in our liver, primarily in our liver and in some cells in our kidneys. Muscles don't do gluconeogenesis. They don't make their own glucose. We'll see how that plays out later. Okay? So what I'm describing to you is happening in the liver, and it's happening in some cells of your kidney. How does the process of gluconeogenesis start? Gluconeogenesis starts with the end product of glycolysis, and that is pyruvate. But I've told you that the cell can't reverse the pyruvate kinase reaction. Instead, it has to do what I call a 2-step around that reaction, meaning it takes 2 different reactions to go from pyruvate back to PEP. The first reaction is one of the reactions that does not occur in the cytoplasm. It occurs in the mitochondrion. It's catalyzed by an enzyme known as pyruvate carboxylase. Pyruvate carboxylase. You can see that it takes ATP and bicarbonate. Bicarbonate is a source of CO2 to produce a 4-carbon molecule known as oxaloacetate. So we're taking a 3-carbon molecule, we are putting putting a carbon dioxide onto it to make a 4-carbon molecule known as oxaloacetate. You'll hear a lot about oxaloacetate next term. Okay? Oxaloacetate is then moved out of the mitochondrion, where this reaction occurs, into the cytoplasm, where the next reaction occurs. In the cytoplasm, there's an enzyme known as phosphoenolpyruvate carboxykinase. You're much more likely to call it PEPCK. PEPCK uses energy from GTP. There's a different triphosphate. Energy from GTP to convert oxaloacetate into phosphoenolpyruvate. We started with a 3-carbon molecule. We burned 2 triphosphates. And by the way, the energy of GTP is exactly the same as the energy of ATP. To get to phosphoenolpyruvate, that's the 2-step that I'm talking about. Okay? Once we're at phosphoenolpyruvate, learning gluconeogenesis is pretty simple. It's the reversal of all the steps that we saw of glycolysis up until fructose 1,6-bisphosphate. There's our friend again. Okay? It's at that point that reaction that would go up to fructose 6-phosphate becomes energetically unfavorable, because in the PFK direction, you might remember, it used ATP to make that reaction go forwards. If we try to make it go backwards, we're going to synthesize ATP, and that's not practical. There's not enough energy here to do that. So rather than try, the cell does something kind of cool. Instead of taking that phosphate off of fructose 1,6-bisphosphate and putting it on ADP, it says, I don't need that phosphate. Let's just clip it off. Clipping a phosphate off is energetically favorable. So there's an enzyme that clips that phosphate off. It's known as fructose 1,6-bisphosphatase, big mouthful of a name. You can call it F16BPAs, BPASE. Okay? So we've now bypassed 2 of the 3 energetically unfavorable reactions in the reverse direction. We're getting ready for the 3rd one. Once we get to fructose 6-phosphate, that phosphoglucoisomerase that we talked about before kicks in, converts fructose 6-phosphate back to glucose 6-phosphate, and then glucose 6-phosphate must be moved to the endoplasmic reticulum, where there's an enzyme called glucose 6-phosphatase that does something exactly equivalent to what fructose 1,6-bisphosphatase does. It clips off a phosphate. It's a lot of stuff. You just learned the whole pathway there in about 5 minutes. Yes, question? Does it matter if it's the rough or the smooth? It doesn't matter at this point for us. Now, I have a song that brings all this together. It's my favorite song I sing. It's one that takes a lot of energy, so I hope you'll bear with me. And I hope you'll sing with me. It's to the tune of Supercalifragilisticexpialidocious. When cells have lots of ATP and NADH2, they strive to store this energy as sugar. Yes, they do. Inside of mitochondria, they start with pyruvate, carboxylating it to make oxaloacetate. Oh, gluconeogenesis is so exhilarating. Memorizing it can really be exasperating. Liver cells require it, so there's no need for debating. Gluconeogenesis is so exhilarating. Glucose, glucose come to be. Glucose, glucose come to be. Oxaloacetate has got to turn to PEP, employing energy that comes from breaking GTP. From there it goes to make a couple phosphoglycerates, exploiting enolase and mutase catalytic traits. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATPs per glucose is the needed energy. Gluconeogenesis is liver specialty. Oh, glucose, glucose, joy to me. Glucose, glucose, joy to me. Converting phosphoglycerate to 1,3-BPG requires a phosphate that includes ATP energy. Reduction with electrons gives us all an NAD, and G3Ps isomerize to make DHAP. Oh, gluconeogenesis is anabolic bliss, reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Oh, glucose, glucose factory, glucose, glucose factory. The aldolase reaction puts together pieces so a fructose molecule is made with 2 phosphates in tow, and one of these gets cleaved off by a fructose phosphatase unless F26BP's acting, blocking pathways. Oh, gluconeogenesis, a pathway to revere that makes a ton of glucose when it kicks into high gear. The cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Oh, glucose, glucose, jubilee. Glucose, glucose, jubilee. From F6P to G6P, that is the final phase. The enzyme catalyzing it is an isomerase. Then G6P drops phosphate and a glucose it becomes inside the tiny endoplasmic reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard because our professor lets us use a filled out index card. Gluconeogenesis is not so very hard. It's true. All right.